So I know you all can hear me. Let me go ahead and share the screen. Okay, can you see the slide?
Yes, I can see the slide.
Okay. Then I'll make it, oh, sorry, I'll make it full screen. So yeah, we're just continuing from last week's discussion of metabolism. I believe this is the last one. Probably a little bit less important this time, but there is probably just as much material here. So I'm assuming you all did the homework, the discrete questions from the 2 chapters in the book. Does anyone have any questions about the homework or questions about what they read? All right. If not, you can ask as we go through the material here. So here's an outline. I know that last time I spent maybe a little bit too much on the content review, which is stuff hopefully you've already read, and that left less time for the discussion, the practice questions on the practice passage. So this time I'm going to go a little bit faster on this. So we're finishing up aerobic respiration. Last time we went through anaerobic respiration, and we went through the steps of aerobic respiration that did not require oxygen. Here we're doing the electron transport chain, which is, hopefully, as you know, the most important step as far as energy and the one that actually uses oxygen. And then the rest is all fats or lipids, a whole bunch here. Go over the basics and then all the metabolism. So the electron transport chain, this is another thing where this is thankfully a lot simpler, I think, than something like glycolysis. There's fewer proteins involved, but you probably should know those, but you should definitely know the basics here, which are hopefully were taught in intro bio. So the electron transport chain occurs across the inner membrane. Crista membrane, remember it's the folded, mitochondria has 2 membranes. This is the folded inner membrane. So those other steps, the Krebs cycle, that was in the matrix, so the inner cavity of the crista. The process we're doing here to make ATP is called oxidative phosphorylation. So you can contrast this, and we call it that because we're using the power of reduced electron carriers, NADH and FADH, to make the ATP. You contrast this with substrate level phosphorylation, which is everything else we saw. So the glycolysis ATP and then the 1 ATP you get out of the Krebs cycle, that's called substrate level phosphorylation. It's just a product of a normal chemical reaction. So the inputs here are those high energy electron carriers. We made these things. We made these in glycolysis, pyruvate dehydrogenation, and in the Krebs cycle, we're making them so we'd have those electrons to use as essentially a source of reducing power for this electron transport chain. And then we need oxygen as the final acceptor, as you're going to see in the diagram. Output, ATP, energy, H2O, which we just breathe out usually. As I alluded to before, this is going to give you most of the energy during respiration. So good numbers to know here. A theoretical maximum of 24 ATP from all the NADH and FADH we already made. You get 3 per NADH and you get 2 per FADH. You'll see why in a minute. And then definitely know these numbers. So for the entire process, for completely breaking down glucose with aerobic respiration, theoretical maximum is 38 ATP. Typical, usual number, if they ask for a math question about this, you'd want to say it's 30 ATP. Here is the overview of the entire process. Hopefully the book will tell you a little bit more about what all these proteins are, but just to go through the basics here, there are 4 complexes. It goes in order from highest to lowest energy, 1, 2, 3, 4. What we're doing here is we're essentially passing electrons. Well, I summarize it here, but whenever we use NADH, so NADH is the better one. So when we use NADH, we start with complex 1, and then those electrons go down and down through gradually lower energy molecules until they go to complex 3 and then complex 4. Complex 4 is the site of the final electron acceptor. And then with FADH, we actually skip complex 1 and enter complex 2, which is lower energy. This is why FADH produces less ATP. And remember, we talked about, well, they call it succinate CoQ reductase. We called it succinate dehydrogenase as part of the Krebs cycle and the electron transport chain, where you can see right here, we're drawing it as one step essentially, because that's what it is. This is a Krebs cycle step, succinate to fumarate. And then there's FAD turning to FADH is not shown, and then those electrons move through the iron sulfur complexes. So this basically just says what I said there. We're taking electrons from higher energy molecules in a stepwise fashion to lower ones, and that energy is used to pump these protons. And then when they diffuse back, they go through a protein called ATP synthase, which is basically like a windmill, or it's like a mill. It has a wheel almost that it turns, and that makes the ATP. Basic regulation here, activated by low concentrations of ATP. Makes sense. We have less energy. We want to make more. Certain poisons will block steps of this, and that's why they kill you. Most famous one is cyanide. Takes the place of oxygen as the final electron acceptor and basically clogs up the whole thing. All right, practice questions. So compared to the matrix, the pH of the intermembrane space when the electron transport chain is active is higher, lower, the same, or you don't know. Feel free to type the answer or shout it out. It's okay if you're wrong. You're here to learn. Or if you need me to push you in the right direction, I can do that as well.
The chat is turned off.
Can you share the question? Because we can't see the questions on.
Oh, you can't see the question? Can you see the questions now?
Yes.
Okay.
No. We still see the last screen you had, the overview.
Oh, I see the practice questions, and the chat is off.
Okay, well, I guess I can't have both. We had this problem last time where the recording wasn't showing. I guess if someone types, since some of you are talking, if someone types something in the chat, can someone just read it to me? Oh, showed you the answer, but can you see the slide now?
Yes, we can see the slide. Okay.
So sorry, I showed you the answer there. So the answer is lower. This is based off of a question I saw during my MCAT practice. So look back to hear what we're doing. We're pumping the protons against the gradient. So we're creating an artificially high amount of protons here in the inner membrane space. High protons, low pH. All right, here's one that's a little bit different. So DNP, which is a banned weight loss drug, works by allowing protons to flow freely across the crystal membrane. Essentially makes the membrane leaky. So which protein of the electron transport chain is this going to render non-functional? If you need to see the proteins again, I can go back.
Yeah, can we see the proteins?
Those are the complexes that has 3 out of the 4 proteins. Can I go back now? Or do you want to stay here?
No, you can go back.
What do you think?
Honestly, I'm going to guess. I was going to choose the second option.
Cytochrome C oxidase. Let's
Yeah.
see. ATP synthase. Which that's the one that was not shown here. Because remember, that's not part. Actually, maybe this isn't good. It's not part of the. It's not one of these proteins here that actually involves electrons, but it mentioned here. So all these proteins here are shuttling the electrons along and pumping the protons. And then to actually make the ATP, we've pumped them against their gradient. So to make the ATP, they diffuse back through ATP synthase. ATP synthase is a channel protein. So these protons diffuse back through that, and the energy from that essentially turns a crank to make ATP. So in that, it goes through there because, remember, plasma membranes are hydrophobic, so protons aren't going to just diffuse through them. But when we take this drug, we're essentially making the membrane leaky, so the protons just flow through the membrane, and they don't flow through ATP synthase. Well, if they're not flowing through that channel, they can't make ATP. All right, here are lipids. We're not going to go through all this. I just have it here. You're going to have these slides, so this is just for your reference. So part of this was to give a basic overview of lipids. Really, we're going to focus on fats, but lipids are going to be covered. Lipids as a whole are going to be covered in the organic chemistry and the biochemistry when we go through all the different biological molecules. But just for now, lipids are kind of they're not like carbohydrates and proteins where they have like a precise definition. Lipids are really just defined by they're hydrophobic. They're not soluble in water. So you get a lot of diverse molecules here, like all of these. But the important one to focus on here are fats. Fat is just, usually when you hear the term fat, it could refer to any of these, but typically when you hear fat, it means triglyceride. A triglyceride is just a polymer, essentially, of fatty acids. So these two are what we're going to be focusing on, fatty acids and triglycerides. So fatty acids are just, from organic chemistry, they're carboxylic acids, but the carbon chain is very long because, remember, they're hydrophobic. And then triglycerides, we put 3 of them together to it's an alcohol that has 3 OHs called glycerol. Saturated versus unsaturated. Again, we'll probably circle back to this, but pretty simple here. So saturation in organic chemistry refers to the number of hydrogens on it. So something that's saturated has all the hydrogens it can take. And it has all the hydrogens it can take because there are no carbon-carbon double bonds. We have a double bond here, but that's part of this functional group. If we didn't have that, then it wouldn't be a carboxyl, it wouldn't be a fatty acid anymore. But in this carbon chain, we don't have any. Unsaturated, you never see triple bonds. But unsaturated, they have varying degrees of double bonds, usually just a couple. The ones you actually see in nature are these, the cis unsaturated fats. Notice that the chain is kinked, which is why they don't pack as tightly. So you'll know, plant fats are a lot of unsaturated fat, and they're liquids. They don't pack as tightly. Trans fats, the double bond is reversed, and you'll notice these actually, in terms of shape, these look a lot closer to the saturated fats than to the unsaturated. And that might help explain why these, are catastrophic for your health and you really aren't supposed to eat any of them anymore. All right, digestion. So whenever we do the biology unit on the digestive system, we will go into the process of digestion for all the macromolecules. But for fats, it's good to cover this here because fats, you'll see, fats are the one that's different than carbohydrates and proteins. So remember, fats are triglycerides. Here's a triglyceride. Three of these chains linked up here. We use, your pancreas actually secretes lipase, which breaks this bond. It's an ester bond here. So it breaks it up and we get what we started with, a glycerol and 3 fatty acids. They do not have to be identical. And then the details here will be covered again later, but this sort of tells you what we're doing with those. So fats are unique because they don't go to the bloodstream directly. Carbohydrates and proteins, we break those down and then the components go into the bloodstream. Here, it's actually part of the lymphatic system. So we made those fatty acids here. They go into the intestinal epithelium, and then we turn them back into, so only the fatty acids can diffuse across, but we turn them back into fats or triglycerides. And the reason we can't put them in the blood is because they're hydrophobic, so they wouldn't mix. So what we do is we put them in these hydrophilic proteins called chylomicrons, and the proteins surround them so they're not touching water. And then those can be picked up in the blood, and those usually go to muscle because we're going to learn muscles will use fat for energy, and then to adipose tissue, fat cells where they can be stored.
Can I ask for a quick clarification on the previous slide?
Yeah, go ahead. Sure.
So it looks like, is it digested at one point and then reassembled?
Yeah, essentially. So it's hard to call it because you'll notice, I mean, this is a very basic step. So we haven't done, this is pretty trivial, right?
Mm-hmm.
It's like, we're going to cover glycogen. If you know anything about glycogen, it's pretty easy to just add and take off those glucoses, right? So it's kind of the same thing here. You'll notice there's no ATP involved in this one way or the other. So we don't care to do this reaction back and forth.
Okay.
Yeah, that's what it is, essentially.
When it's first entering, like on the next slide, it's, so you have fatty acids. And so when it enters like the digestive system, is it being broken down into those components at that point?
So it's in your stomach and it's in your small intestine, right? So you eat something that has these, right? You don't eat fatty acids, you eat triglycerides or fats. So you eat these, and it's going to be in your stomach, and then it's going to go to your small intestine, and your intestine and your pancreas secrete lipase. So so this is dissolved in it's dissolved in with all the other food in your intestines, and then this is secreted. So you have this floating around, and then you have this floating around, and only the fatty acids are then taken into the cell.
Okay, thank you so much.
All right, no problem. So oxidation. So this is when we use them. We don't like to use fats as energy, but we will if we have to. So we already said here, we already make first step, not oxidation, but the first step of digesting them is turn them into glycerol and fatty acids. Glycerol, we learned last time, can actually have, well, it can do both. So glycerol can input into gluconeogenesis and make glucose, but it can also be converted into glucose 6-phosphate and enter glycolysis. Just depends on what our energy needs are. The most important, the only way to break down fats you need to know for the MCAT is called beta oxidation. This occurs in the mitochondrial matrix, same place as Krebs cycle. You're going to see an overview on the next slide, but the products of this that are important to know, it produces acetyl-CoA, which we know the main use is to enter the Krebs cycle. We're going to see another use in a minute, which, remember, acetyl-CoA cannot enter gluconeogenesis. And then we can also produce these high energy electron carriers from before, NADH and FADH, which then make more ATP from the electron transport chain. Probably already knew this, but they do ask questions about this. Fats give more energy per gram basis. On a per gram basis, fats give more than double energy than carbohydrates and proteins. And there's two good reasons for this that are good to know. So remember, when we're getting energy from food, we're oxidizing it. We're oxidizing the molecules. Well, fats are, I mean, look at these molecules. It's mostly just an alkane. So this is a very reduced state. So we get a lot, when we oxidize it, it's a longer reaction that produces more energy. And also we said they're defined by being hydrophobic. Well, if they're hydrophobic, they're not surrounded by a water shell. So carbohydrates, when they're in your body, are surrounded by a shell of water, like those ions, if you remember from like Gen Chem. The fats can pack very tightly together in your fat cells because there's no water to surround them. Here's an overview. Again, probably not super important with this one to know all the enzymes, but definitely the products here. So the important thing to know is that it's a cyclical process. We're digesting this thing 2 carbons at a time. So the first step, we have the fatty acid from before, OH. There's no CoA anywhere. It's just OH at the end. First, we have to activate it with coenzyme A. You're going to see that. You see that a lot in biochemistry. It's the same as, acetyl-CoA. So we need an ATP to activate it. And then that's what's entering here. And through this stepwise oxidation, we make one FADH and one NADH, and we eventually chop off a 2-carbon chunk, or acetyl-CoA. Good to know that that's a 2-carbon thing. And so you're going to see this, again, the exact steps aren't super important, but it is interesting to look at. You're going to see this sort of stepwise oxidation a lot. So we go from just a single bond to a double bond to an alcohol to a carbonyl, and then we chop it in half and add another carbon. We chop it in half. So each step we're getting more and more oxidized. And then we now have one of these activated fatty acids that's shortened by 2 carbons. We just enter it back in the cycle. And this will just keep feeding it back in until at the very last step it'll be a 4-carbon chain, and we'll cut it in half and make 2 acetyl-CoAs, and then we're done. And that's for saturated. So you'll notice that this was a saturated chain. Unsaturated, a little bit different.
I have a question. Real quick. Did we go over chapter 10?
Sorry, what was chapter 10 was the electron transport chain, right?
Oh, okay. And the citric acid cycle.
Citric acid cycle was last week.
Oh, okay. Thank you.
Yeah. Yeah. If you feel like I've missed anything, you can let me know though. Back here. Okay. Unsaturated, a little bit different. So notice how when we chop off these 2-carbon chunks, we're doing it at the end that had the carboxylic acid, right? So nothing happens till we bump up against this. So like here's an example where the bond is in the middle. Well, first we're going to chop off these chunks over here by the CoA, So nothing matters until we get right up on it. And then right here, well, that's no good because if we chop this, we're going to have the alkene at the end. So everything's going to kind of fall apart. So we just use this enzyme here to, it's an isomerase. So it just remember, if we move a double bond, we've made an isomer. So it just moves the double bond over. And now look where we're at. We're at this second step here. So we just skipped out. So basically we've just substituted the first step for this isomerase. And then we're now at this step where we have the double bond. And then we just go through it like normal. It's also a little bit different and a little bit more complicated if you have a fatty acid chain that is odd numbered. Because remember, we're chopping off 2 carbon chunks, so if it's an odd numbered, then it kind of messes everything up. But that's, again, the details of that aren't important here. Ketone bodies is the other thing we can do with acetyl-CoA, and it's really just a longer way to the same thing of throwing it in the Krebs cycle. So hopefully by now you've caught on. Glucose is the primary energy source of the body. If you eat, fats, sugars, and proteins, we're going to prefer to use glucose for energy and use the fats and use the proteins for building blocks for other things. So actually, typically your brain runs only on glucose. Glucose can cross the blood-brain barrier. And even if you're totally starving, there's actually a certain amount of glucose your brain has to have or it won't work. Even if everything else, we can use beta oxidation to get energy from fats, and you'll see later we can get energy from proteins as well. But there is a minimum that you have to have some glucose. However, when we're burning fat for energy, we probably don't have, a bunch of glucose laying around. So we can't use that for energy, but burning fat, we're probably not doing that, in the brain where it's needed. So when we make this acetyl-CoA, we usually don't move ATP around between cells. ATP is used for energy within the cells. So we can move the acetyl-CoA around, but there's a problem. Acetyl-CoA can't cross the blood-brain barrier. So what we do instead, if we're running on fats and we're making a bunch of this acetyl-CoA, we will turn it into ketone bodies, which is done by your liver. There's 3 ketone bodies to know: acetone, acetoacetate, and D-beta hydroxybutyrate. And these are synthesized in a few steps from acetyl-CoA, 2 carbon units. Acetone, we don't use it, we just breathe it out. However, these two, they're hydrophilic molecules, so they can float around in the blood, and they can cross the blood-brain barrier or go anywhere else they're needed. And once they're in the cells, in the cytoplasm, we can just go back to acetyl-CoA. So this is like a currency for that. We can just go back and forth between acetyl-CoA and these ketone bodies. So that's ketosis. So if you're starving or if you're intentionally, like a ketogenic diet, you're going to have a lot of these because you're not eating carbohydrates. So we need this instead to move around for energy. All right, first practice question. Again, you'll have to say the answers or read someone else's answer. So how many times would beta oxidation occur to completely break down a fatty? So we have a fatty acid that is 8 carbons long. How many cycles of that beta oxidation do we have to do? Let me know if you need a hint with this one.
Does it require us knowing whether it's saturated or unsaturated? I guess not, but
No, it does not.
Okay.
You can give us a hint.
Okay, so remember, beta oxidation breaks down fatty acids into acetyl-CoA, which is a 2-carbon chunk. So how many times do we have to chop off 2 carbons if we start with a fatty acid that's 8 carbons long?
So wouldn't it be 4?
Well, not 4, because we don't have, because remember, what's the final step? The final step, we chop a 4. So let's go through it in your head. So we start with 8 carbons, chop off 2, that's 6. That's step 1. Then the 6 goes back, chop off 2, that's 4. Then chop the 4 in half, and that's 2 2s, and we're done.
So it's 3.
3. Yeah, so always with beta oxidation, the final step, it's not shown here because this is just a generalized, but the final step is always going to be a 4-carbon that we then chop directly in half, and we get 2 acetyl-CoAs, and then we're done. All right, this one, you have to know all the numbers for this, so let me know if you need a hint, and I can give you some of the important information with this one. How many net ATP, so once we feed everything into the electron transport chain, how many net ATP do we get from one cycle of beta oxidation? So when we chop off that acetyl-CoA once, how many ATP do we get?
So I studied I read chapter 10 and like the citric acid cycle. I didn't even get to chapter 11. So I'm just like, I know from the citric acid cycle, you get 12.5 ATP net, but now I don't know, like.
Okay, wait, sorry, I should have said the maximum amount. So yeah, you're right. So we get an acetyl-CoA that we then feed into the citric acid cycle. So 12.5, I was using a higher number. So the numbers aren't really important with this one, but the important thing here is that you know every step we're getting ATP. So can you trace all the ATP we'd be getting from one cycle of beta oxidation? So here is the overview. So there is FADH. Do you remember how many ATP do you get for one FADH? Maximum.
1.5.
Yeah. Well, I said 2. 1.5 is more realistic. So yeah, 1.5 would be acceptable. And now what about NADH?
2.5.
Yep. 3 maximum, 2.5 probably actionable. So we get one of each of those, and then we get acetyl-CoA. How many do we get for one acetyl-CoA? Which it becomes 1 turn of the citric acid cycle.
Is it 12.5?
12.5. You know, that's the average. And that comes from 3 NADH. These are exact numbers. You get 3 NADH, 1 FADH, and you get an ATP just already made. So if you multiply all that out, the maximum would be 17, but it would be less than that. The reason it's less is because NADH is actually not, so, you know, we probably have to move NADH across membranes at some step in this. NADH does not actually go through the membrane. Instead, what happens is there is a transport protein that basically it basically just moves the H across to an NAD on the other side. So the membrane transport steps that you don't see, you know, whenever we're making ATP, waste some of the energy. All right, I think that's okay.
I have a question. Can you just go back really quick?
Yep.
So, So beta oxidation to make acetyl-CoA, right?
It's to make energy, and the way we do that is we bust up fatty acids into acetyl-CoA, and we get some free FADH and NADH as well.
So if one acetyl-CoA makes 12.5 ATP, and we have 4 from beta oxidation. Well, I'm sorry. I think I'm mixing up the last.
You're mixing up this is one cycle.
I see.
One, yeah. So I'm not saying any specific fatty acid here. I'm just saying we're starting with some fatty acid. We're doing all these steps just one time. So we get the 12.5 here, and then we get these extra ones that also feed in.
Oh, okay. I see. Mm-hmm.
You good?
Yep, I got it.
All right. So yeah, now you start to see why fats give us a ton of energy, because we got 17 ATP max from not one fat, but just one little step of breaking it down. And fats have 3 of those fatty acids per each molecule. Oh, all right. Synthesis, a little bit less important here, but big thing here, the overview mentioned talking about non-template based synthesis. So when we talk about, proteins, we use mRNA as the template, and nucleic acids we use nucleic acids, when we're making RNA and DNA, we use the other strand of DNA as a template. This is not template based, and neither is, we're going to see, glycogen, not template based. This occurs in the cytosol, not the matrix, and it's not like gluconeogenesis. It's not just the reverse of beta oxidation. The enzymes are basically all different. The energy input is different. Remember, from the pentose phosphate pathway, NADPH. This is when we're using it. And malonyl-CoA is the building block, and the intermediates are different. So, but like last time with oxidation, we cut off 2 carbons. Here we're going to add 2 carbon fragments stepwise. And we're going to make fatty acids. And remember, it's pretty easy to turn those fatty acids into triglycerides. We can also turn them into phospholipids, which you're going to see in a later unit are very, very similar to triglycerides. Here's the steps. So notice that the oxidations, the redox reactions that are occurring are the opposite of last time. So we're combining 2 carbonyls, then we make an OH, we get rid of it, make a double bond, and then we get rid of the double bond. But notice how the building block is not, it's not acetyl-CoA, it's malonyl-CoA, which is 3 carbons, but we only add 2 carbons. That's because the last carbon gets lost as CO2. We essentially add CO2 to make this thing, and then it gets lost. Glycogen, this one's pretty easy. Glycogen, probably know, glycogen is like starch, but for humans. So it's used as a glucose storage mechanism. It's less permanent than fat. So if we have a lot of glucose lying around, we can make this thing. And unlike starch, it's very, very well branched. If you look up a picture of it. Here's what happens. We went over 2 weeks ago, we went over what the different influences are when we want to make this thing, and when we want to break it down, aka glycogenesis and glycogenolysis. So it is a polymer of glucose, but the important thing to get from this is that we're adding phosphorylated glucose. So look at the steps here. So glucose 6-phosphate, remember from last time, that's hexokinase that makes that. And then we can just use that for glycolysis. We can use it for the pentose phosphate pathway as well. But if we want to put it on glycogen, we've got to move, we make an isomer. We move the phosphate over to the 1 carbon, and then UDP, UTP, and UDP. So remember ATP and GTP, it's just another energy carrier, but the nitrogen base is different. And then the UDP attaches it, and then that is used to put it onto glycogen. So glycogen is just this big glob. We're just adding and removing molecules from the ends. And we also intentionally branch it. And then if we want to get rid of it, we can use The opposite, glycogen phosphorylase and glycogen synthase, good enzymes to know. We just pull off glucose, but remember, glycogen does not have phosphates in it. So we have to add the phosphate when we pull off glucose 1-phosphate. All right, let's go through these quickly so we can do the passage. So after eating a large sugary meal, which process will predominate? So if we have a lot of glucose laying around, which one are we going to do?
Would it be the first one, glycogenesis?
Yes.
Okay.
We have extra glucose, so let's store some of it. I'm just going to tell you the answer to this one. It's true. So malonyl-CoA, remember we make that from acetyl-CoA plus CO2, and it's irreversible, and this is a reason we can use this step to essentially separate out acetyl-CoA that we want to use for making fats. When would fatty acid synthesis be upregulated?
Wouldn't it be during starvation?
Well, during starvation, if you're starving.
Oh, you know what? I'm so sorry. I'm thinking of something else. I'm so sorry.
So 3 of these answers are really actually saying the same thing, and therefore cannot be correct. So I'm just gonna, so we can get through this and do the passage. So yeah, during starvation, we want to be using the fats for energy and not making more. So synthesis and breakdown, we never want them to happen at the same time. So if one's happening, the other's not happening. If glucagon is high, that's because there's low glucose. So we want to make, that means we're making more glucose. So we probably, well, you could make a case for that one actually, but definitely not this one, ketosis. So ketosis occurs during, ketosis occurs when we're doing a lot of beta oxidation. So if we're doing beta oxidation, we're not doing this. But if we eat a lot of carbohydrates, it means we have a lot of energy laying around. And we might not turn the carbohydrates into fat, but we may use the fatty acids we may store, or we may use other acetyl-CoAs from proteins or from carbohydrates to make fatty acids. But yeah, this is not a good question. You could argue for the third one as well. All right, here's the passage. So I'm going to stop sharing and give you the link. Let me know if that doesn't work for you. Take about 10 minutes for this. Let me know if you think we've had enough time and we can go through the answers.
For some reason, I can't access the chat. It says chat and channel meetings is only available to team members.
Anyone else having the problem?
Me too. I think it might be because I'm logged in as a guest.
That would be it, but that shouldn't be here. Can you see it in the global chat?
No luck, but I can always catch the recording after.
Well, if you see, you click the,
Yeah, I don't even know how to access that like chat. Sometimes it opens when I open Microsoft Teams, and sometimes it just doesn't.
Let's try something more creative here. You can see when I share the screen though, right?
Yeah, can you just like enable the chat for this meeting? If that's
Well,
possible.
I don't know how to do that. So here's my idea I came up with. Can you see the slides?
Yes.
Yeah, you can just open it like that. Yeah.
I made a short link for you, so type that.
Thank you so much.
Sorry, I'll figure out how to give the chat. All right. Does everyone have it now?
Yes.
Okay. Stop sharing then. All right, how are we doing? Are we about done, or which question are we on?
I'm finished.
We're done.
I'm done.
Anyone still working? All right. Just gonna leave. Let me know if you can see the screen. You see the screen now?
Yes.
All right. So questions. So number 1, all the following. Let me pull the passage up. So if we have the CPT2 deficiency, remember we need this to move long chain fatty acids. So often the best strategy with MCAT questions is to just eliminate all the wrong answers. So what will be beneficial? So if we can see it, well, small and medium chain, we're good because it said that those don't need, it said in the passage that those are transported without CPT2. But, and then if we eat frequently, we'll have less of a need for fatty acid metabolism. However, if we're fasting, so if we're eating a large meal, what's going to happen is it's going to be like a cycle. You're going to eat a huge meal, you're going to store glycogen, and you're going to store fat. And then when you're fasting, or when you're starved for energy, you're going to need to break down fats. So then you're going to run up into this roadblock and you're going to have problems. Question 2, levels of carnitine in individuals with the deficiency. So CPT2 we saw is this blue one here. So it makes carnitine. So if it's deficient, then you don't make as much, so it'll be decreased. Why do some patients with CPT2 deficiency suffer from hypoglycemia? Did anyone get this right and want to explain it? I thought this was the hardest question.
I got it right. So I learned this in Biochem 2. So I know that glucogenesis is like you're producing, I think you're trying to produce I'm going to say it wrong. Glucagon, I think that's how you say it.
You're producing glucose.
Right.
Your glucagon upregulates gluconeogenesis, though.
But so the reason I got that one because I did process of elimination. So I knew that it couldn't promote insulin secretion because for some reason I just assumed, well, from reading it, I kind of just assumed that the patient who had this deficiency wasn't really consuming anything. Like in the paragraph below the chart, it said that like they had long periods of fasting. So when you're fasting, you're not going to be really producing insulin because you're not getting you're not eating. When I'm thinking insulin, I'm thinking you're eating. When I think of glucagon, you're fasting. So that's why I took that one out.
Yes. Yeah. So it's good. You can keep going, but I'll just say it's good that you're doing process of elimination. So. Yeah, you can just simply say, so you're right. Your last point really explains why this is wrong. Insulin is produced when you've eaten, and fatty acids. So usually we like to use fats. We don't like to use fats for energy. So we only do this when we're starving, essentially. So there would be no insulin.
Right, and then acetyl-CoA. Why did I say it? I think I said that one was not it because. Again, why did I say it? I don't know. My brain went to glycolysis and it just ran with it. So I was just like, I don't think glycolysis is going to happen if you're not eating anything.
Well,
so I was like, okay.
Yeah, I can go.
So, well, I said this one's the right answer to the wrong question. Do you want to go? Sorry, you want to say something?
Oh yeah, this is Rashina. So I was gonna say that well, first you need to know like what hyperglycemia is. So it's like when your blood glucose levels is low, so just low glucose in the blood. And I think fatty acid oxidation does yield acetyl-CoA, but it's a better answer. so fatty acid oxidation fuels glucogenesis, because with CP2 it doesn't make the carnate protein, which transfers acetyl-CoA from the cytosol to the matrix, which, you know, fuels like the citric acid cycle and then other things like the glucose cycle and stuff like that. So if, you know, if we don't have acetyl-CoA, which is created from fatty acid oxidation, we can't create glucose by gluconeogenesis. So, but if you don't have that, the CP2 protein or enzyme, you know, you can't fuel gluconeogenesis. So that's why D is right. I don't know if that makes sense. That makes sense.
Some of that, you're on the right track with that. So, like I said, this is the right answer to the wrong question. So we should know from the previous slides that fatty acid oxidation does yield acetyl-CoA. We're chopping up fatty acid into acetyl-CoA. But something to keep drilling in is that acetyl-CoA cannot feed into gluconeogenesis at all. That's why we made the ketone bodies as the energy store. We cannot make glucose from acetyl-CoA. So really, this answer really is wrong because it has nothing to do with the question. Because it has nothing to do with glucose, because you can't make acetyl-CoA. You can't make glucose from acetyl-CoA. You can only do the reverse when you're doing the citric acids, when you're doing pyruvate dehydrogenase. And then choice C is completely wrong because we saw there was no glucose involved in that reaction. And even if we want to take it further, we already said we can't make any of the things we're making from fatty acids, we cannot turn into glucose. So I said it before, I thought this one was hard because the correct answer, you could get this one right if you crossed all those out, but this correct answer, I did not cover this. I don't know if the book did, but this is just a biochemistry fact. Because, well, first of all, to do fatty acid oxidation, we had to have we start with a fat, and that had glycerol that can feed into it. But fatty acid oxidation, typically, if we're doing gluconeogenesis, we're not breaking down glucose, right? Because we wouldn't make and then break down glucose at the same time. It would be a waste. So if we're doing gluconeogenesis, we're using fat for our energy. And fatty acid oxidation produces ATP at the end, and we need ATP to do gluconeogenesis. So the acetyl-CoA is used acetyl-CoA does not make glucose, but it does make ATP at the end, which is used as the energy source for gluconeogenesis.
And gluconeogenesis is the formation of glucose?
Formation of glucose from, if you want to treat it as the reverse of glycolysis, you can say it makes glucose from pyruvate, but it can make glucose from a lot of stuff. But importantly, not acetyl-CoA.
Got it. Thank you.
A deficiency in what other enzyme would most closely mimic the clinical manifestations? So the clinical manifestations of this enzyme being deficient were when we ate fats, everything derailed. We couldn't break them down. So it helps if you know what all these other enzymes do. We could just, again, cross off the answer choices. Did anyone want to explain this one, or?
So I put C, pyruvate carboxylase. I don't know if pyruvate carboxylase is the enzyme in the PDH pathway. And then, you make acetyl-CoA from pyruvate, but I don't know why it's B.
So again, if we know what all these do, so glucose 6-phosphatase, well, that one tells you what it does. That's gluconeogenesis. That's the final step of gluconeogenesis. Fatty acid synthase, that's part of fatty acid synthesis, which is the opposite of this reaction. So really, that's totally tangential. And then pyruvate carboxylase. So pyruvate, we didn't see pyruvate anywhere in the fat pathway. either making or breaking down fats. Pyruvate carboxylase, well, first of all, pyruvate, that is the product of glycolysis and the input of gluconeogenesis. So pyruvate carboxylase is actually another step in gluconeogenesis. So all the answers here are crossed off because they have nothing to do with fat metabolism. Hormone-sensitive lipase, we didn't cover hormone-sensitive lipase, but lipase, we're going to see again with digestion more, but if we go, I don't want to scroll way back. But if you remember from the first step of, so we don't eat fatty acids, we eat triglycerides, and the first step was to break them down with lipases. So if we don't have lipase, we're also going to clog up breaking down fats. Just like this, if we don't have this CPT2, we're frozen up at this step of fatty acid breakdown. If we don't have this, we're going to freeze up at a much earlier step before we even make the fatty acid. And then this one, anyone want to explain?
So I said it would worsen a patient because if you go back to the last paragraph in the passage, it talks about a symptom is often exercise and fasting. So to me, as soon as it said fasting, I'm thinking of glucagon, because when you're fasting, I just assumed, I put those two together. So if you're administering more, it would just make it worse instead of making it better. I would think if you administer glucose, it would make it better. So that's the reason why I chose that answer.
Yeah, actually, that is not how I answered it, but that is completely correct.
Okay.
And that's good. And that is good. So the way you answered it, you did not need any external information. That's something a lot of these passages, the information will all be there if you just kind of think it through. So that was something where all you had to know was glucagon equals starving, which hopefully you all already knew, and you could get it that way. But really, I think the way, if you want to do this from more of a content perspective, it's because of the effect glucagon has on fat metabolism. So glucagon is released when we're starving. Remember, if we're starving, we're trying to conserve our glucose. So we're going to bust up glycogen so we want to increase our glucose. So we're going to do gluconeogenesis to make it. We're going to break down glycogen, but we don't want to waste it all with glycolysis. So we're going to use fats instead for energy. So if we administer glucagon, we're favoring beta oxidation. Well, CPT2, as we just saw, screws up beta oxidation. All right, are there any more questions about the questions here or about anything else here? I know we're a little bit over time, but I can stay for if anyone else has any questions.
No, I think that I did have a question on the last. So what does glucagon do?
Just in general?
In general, a short summary.
So you really, when you're talking about glucagon, you want to talk about insulin, which, insulin's the one we all know. Glucagon, you want to think it's glucose sparing. That's its effect. So it's released. So insulin, we know insulin gets released when we have too much glucose in the blood, right? Hopefully. So glucagon's the opposite of insulin, essentially. Glucagon gets released when we don't have enough glucose in the bloodstream. That's what triggers it to get released. So it's glucose sparing. So we want to flood the body with glucose. So everything it's doing, the goal is to flood the body with glucose. So the specific things that it does, it increases glycogenolysis because we're going to bust up glycogen to make glucose. Again, increase glucose. We're going to increase gluconeogenesis to make glucose from any of those feeders into gluconeogenesis. So with that, we're going to inhibit glycolysis. And what's important for that question that we just saw if you're inhibiting glycolysis, that means we're not breaking down carbohydrates for energy. So we have to use something else for energy. So with that, we're going to increase beta oxidation. And if we're breaking down fats for energy, we don't want to make more of them, so we're going to inhibit fatty acid synthesis. Make sense?
Yeah, and then so why would administering glucagon worsen the patient's symptoms?
Because so if we're trying to spare our glucose, we're going to inhibit glycolysis. So if we're flooded with glucagon, we're not going to be using carbohydrates for energy, which therefore means we have to use fats for energy. Well, the whole problem with that deficiency was that it screwed up beta oxidation. We were not able to oxidize long-chain fatty acids. So making us more reliant on fatty acids for energy is going to make things worse.
Oh, okay. And we can't produce fatty acids or, transfer to the to the mitochondria. Okay. Okay. I'll sit on it. Thank you.
All right, if there's nothing else, I'll be at office hours this Saturday at 8 p.m. if you all want to look at the slides or sit on this some more and have any more questions or about the homework or anything else. Other than that, thank you, everyone.
Thank you. Good night.
I'm sorry, just one more question. So if you administer glucagon, right, like it's to create glucose, you can't use glucose for, like, I don't understand why. I feel like I'm probably missing something and I should read, but I don't understand why it would worsen the patient's symptoms. Like, what does glucose directly do that relates to fatty acid oxidation?
Well, and you're kind of getting, that's why some of these hormone questions are tricky, because it's all about homeostasis. So what would actually happen is you'd be releasing other hormones to then counteract the effect. But essentially, so we're talking about glucagon and glucose. So yes, glucagon. So you're saying, well, we're making a ton of glucose. Why can't we use it? The reason why we don't use it is because that would then contradict the whole purpose of making it, right? The body is never going to do two opposing processes at the same time because it's just a waste. So if we're doing glucagon is trying to flood the body with glucose, so we will get it from glycogen and we'll get it from gluconeogenesis, but then we don't want to use it all up because then we will have just wasted all that work. So instead, we're going to say, well, we don't have to use glucose for energy. We can use fats instead. We have those laying around, right? So we're going to use those instead. But we made the glucose because we said before, like the brain has to use glucose for some things. So we now have the glucose for the things that require it. And once we're not starving, we have the glucose to use. Did that answer the question or no?
But yeah, I think I should just probably Google it because I don't understand why though. Like, would it worsen the? I mean, and then I guess you would say, like, why would it make it better, or like, what does it have to do with the? Like, I think I need to read because I read for today and I read the citric acid cycle, but I also missed last week. And was last week glycolysis?
Yes.
Yeah, so I missed that. So when you do glycolysis, you make glucose, right? And then does that
No.
Right.
When you do glycolysis, you break down glucose.
Oh, you break down glucose to
gluconeogenesis makes glucose.
Okay.
Which is what glucagon is encouraging.
Okay. So you do glycolysis. So does glycolysis come before or after the citric acid cycle?
But the reason why it makes it worse is because So a fact to know, when we're doing like the endocrine system, you'll learn like the effects of each hormone. Glucagon encourages beta oxidation. It's a direct relationship. beta oxidation was broken by this.
Oh, I see. Okay, yeah, that answers it, right? So it like whatever directly promotes beta oxidation.
Direct,
it can't, yeah.
Yeah, it's not like some side thing. It's one of it's if you look at like a list of the things glucagon does, it's one of the direct things. It encourages beta oxidation and it stops fatty acid synthesis. Again, they're opposite processes. We don't do them at the same time.
Got it. Okay, so obviously it would worsen the symptoms if it's trying to promote beta oxidation, but we cannot do beta oxidation. So, yeah, there's something going wrong.
Yeah.
In the body. Okay, got it.
All right.
Yeah, that's it. Thank you.